We're going to restart. And what we're going to look at is the light-dependent reactions. So we're going to pick up kind of right where we started from. So this first part is the same as we talked about before. Notice these electrons with extra energy are called excited electrons. If they just have their normal amount of energy, they're said to be in the ground state. So before we pick up with what's going to happen with this electron when it leaves the primary electron acceptor, let's go back to some other molecules that we find in that thylakoid membrane. So let's talk about a few other types of molecules. So we have some electron transfer proteins. And the electron transfer proteins, their name's a pretty good description of what they do. They're going to pass this electron around that comes into the system from the primary electron acceptor. And again, they have specific names: plastocyanin, plastoquinone, cytochrome B6 complex, ferredoxin, and NADP reductase. You don't have to necessarily worry about all the names, but it's just easier to call them by their real names and say protein A, B, C, D, E or something. These are going to pass electrons around, kind of like if you ever played hot potato when you were a little kid. Now remember, at every pass, think about what we just said. Every time we pass the electron from one protein to the next, it's going to lose a little bit of energy. There's another protein in the thylakoid membrane called ATP synthetase. And ATP synthetase, notice ASE always tells us what? Something is a protein that's working as an enzyme, is going to synthesize ATP. So ATP synthetase is going to take ATP and it's going to make ATP from the two reactants of ADP and a phosphate group. And usually for a phosphate group, we just abbreviate it with a P with a circle around it. We don't like to write a plain P because that's phosphorus. And then finally, we have a third molecule, or actually a group of molecules called the oxygen evolving complex. And the oxygen evolving complex are these little kind of blue ovals. And remember when we talked about the plasma membrane, we said that you could have proteins that were stuck right on the surface that were called peripheral proteins. So these would be examples of peripheral proteins. And the oxygen evolving complex takes water and splits water into three parts. It takes two water molecules and it combines the oxygens from those two waters to make O2, oxygen gas. So notice this is where the oxygen that gets made as a product is created, at this oxygen evolving complex. And then it takes the hydrogen atoms and it separates. Remember, hydrogen has one electron and one proton. It separates the electron from the proton. So we get an electron by itself. Electrons have a negative charge. And we get a proton by itself, and protons have a positive charge. So we have pigment molecules, which are organized into photosystems. And on this diagram here, the photosystems are the green blobs. And that's the oxygen evolving complex, are these little blue circles. The ATP synthetase are these big, I don't know what they look like, flowers, cupcakes, I'm not sure. And all these purple and pink and orange or various parts of the protein transfer, the electron transfer proteins. So let's think about looking at cyclic photophosphorylation first. So let's look at a diagram of what's happening. So notice here's P700, or photosystem 1 is another way to think of this. And this dark green represents our primary electron acceptor again. Here's our first chlorophyll A molecule in the reaction center. All these other kind of jigsaw puzzle looking light greens, these are other pigment molecules. So what happens, remember we said that that excited electron from the primary electron acceptor, the primary electron acceptor is very sensitive to its electrons having any energy. And so when this electron gets some extra energy from the first chlorophyll A, the electron has so much energy, it just jumps right out of the molecule. And it's captured by one of those electron transfer proteins, one called ferredoxin. And ferredoxin passes the electron to plastoquinone. And plastoquinone passes it to the cytochrome B6 complex. And the cytochrome B6 complex passes it to plastocyanin. And plastocyanin does what? It gives the electron back to the primary electron acceptor. So remember when we talked about cyclic metabolic pathways, we said the key thing was at the end, we got back what we started with. So notice here that primary electron acceptor in P700 lost an electron. The electron gets passed around through a bunch of proteins and the electron ends up back where it started. So that's why this is cyclic photophosphorylation. And notice over here on the left, they have this orange arrow showing you that the electron is losing energy each time it gets passed from one transfer protein to another. It's got more energy when it's in ferredoxin, because ferredoxin is up here, than it does in plastoquinone, because plastoquinone is down here. And when it gets back to the primary electron acceptor in P700, it's got the same amount of energy that it did before it got excited. So we have some electrons that gain some solar energy. They pass it through the antenna complex. When it gets to the primary electron acceptor, the electron in that molecule jumps right out of the molecule, gets passed through the electron protein transfer proteins, and ends up back where it started. Now, when this happens, notice that we end up making some ATP. And we'll talk later about how the ATP is made. But before we do that, let's look at non-cyclic photophosphorylation. So this is a linear metabolic pathway. And here's a diagram of this. This is a little more complicated, so we're going to divide this up into three processes. Photosystem 1 is over here on the right. So first we're going to talk about what happens in photosystem 1. Then we'll talk about what happens in photosystem 2, which is over here on the left. And then we'll talk about what happens in the oxygen evolving complex, which is down here where we see the water. So let's think about photosystem 1. Photosystem 1 basically starts out exactly the same as it did before. Notice this lighter purple in the middle represents our reaction center. So this dark green is the first molecule of chlorophyll A. This light green is the primary electron acceptor. All these green circles along the side, these are some of those antenna complex molecules. So a photon of light hits the antenna complex. Electrons in some of these molecules absorb some solar energy. They pass the solar energy around, and the solar energy eventually gets passed to the first chlorophyll A in the reaction center. That gives it to the primary electron acceptor. And then the electron jumps completely out of that molecule. So that's that yellow line going over here to the right. Now, when it jumps out of the molecule, again, it jumps into that first electron transfer protein called ferredoxin. And the difference is what ferredoxin does with it. Let's go back to cyclic for a moment. Notice that in cyclic, ferredoxin passes this electron to PQ, plastoquinone. Here, PQ is over here. Ferredoxin doesn't pass the electron that way. It passes it to something called NADP reductase. So NADP reductase reduces NADP. Remember, oxidation reduction reactions. And something is reduced when it gains hydrogen. So NADP plus picks up hydrogen and becomes NADPH. So without knowing all the formula for NADP, you can tell that NADPH has one extra hydrogen. So in this reaction, NADP is being reduced. And when it picks up this hydrogen, it also picks up the electron that came out of this primary electron acceptor. Now, what is NADPH going to do with this hydrogen and this electron? It's going to carry them over to the light-independent reactions, to the Calvin cycle, so that they can be used to what? To make carbon dioxide into glucose. So this is going to carry the hydrogen over, and we're going to add these hydrogens to carbon dioxide to convert it into glucose. But notice, chemically, this is a problem, because if we lose an electron from photosystem 1, photosystem 1 has a positive charge now. And remember, we said opposites attract, positive charges attract negative charges. So if photosystem 1 has a positive charge, it's going to be harder to get photosystem 1 to keep giving away electrons, because it's got a positive charge now, so it's going to be holding on to its electrons tightly. Opposites attract. Well, this is where photosystem 2 comes into the picture. At the same time that photosystem 1 is getting hit by light and giving away an electron, photosystem 2 gets hit by light. And really the same thing happens in photosystem 2 that occurred in photosystem 1. When light hits photosystem 2, an electron in one of the pigment molecules in the antenna complex absorbs some solar energy. This energy gets transferred from electron to electron until it gets to the first chlorophyll A in the reaction center. And then that chlorophyll A, again, gives its electron to this purple square, to the primary electron acceptor. And the primary electron acceptor gives its electron to this thing, this purple thing. Well, this purple thing is actually, let's go back a moment. That purple thing is plastoquinone. And when plastoquinone gets this electron, it passes it to the cytochrome B6 complex. That passes it to plastocyanin, and that gives it back to photosystem 1. So we had a problem. Photosystem 1 lost an electron. Okay, we've corrected that problem. We've given it an electron from photosystem 2. But now we have another problem. Photosystem 1 is good now, lost an electron, but it picked one up. But it got its electron from photosystem 2. So now photosystem 2 is missing an electron. So where are we going to get it? This is where the oxygen evolving complex comes in. The oxygen evolving complex splits water into 3 parts. So oxygen, O2, and remember what I said, the plant cell might use this oxygen, but most of it is going to be released into the air. Some hydrogens with positive charges, and electrons. Electrons. So these electrons are going to go into photosystem 2 to replace its missing electrons. So photosystem 1 loses electrons to NADPH, but they're replaced by electrons from photosystem 2. The missing electrons in photosystem 2 are replaced by electrons that came from water. Okay, so how are we going to replace these missing electrons from water? We're not. This is why you have to constantly give a plant water, because the water is used up. Notice after the oxygen evolving complex gets done with it, we don't have water anymore. The electrons were completely separated from the oxygen atoms, and they were also completely separated from the hydrogens with a positive charge. So this is why you have to water your plants regularly. If you don't, they can't carry out non-cyclic photophosphorylation. Now notice we say here that electron transport provides the energy for ATP again. So remember back up here, we also had ATP. And we said we would talk about ATP when we got to the third process, which is chemiosmosis. So let's think about chemiosmosis. And notice this is something that happens during cyclic and non-cyclic photophosphorylation. And it's going to take ADP and what's called an inorganic phosphate, because it's just a phosphate group all by itself. So remember, for something to be organic, it has to have some carbon atoms. This has the formula PO4, sometimes HPO4. So chemiosmosis is going to take ADP and phosphate and combine them to make ATP. Why does that matter? Well, remember, ATP is what cells use for the energy to do everything they want to do. So it's crucial in that respect. So let's look at what happens. So here we have photosystem 1 in the green. We have photosystem 2. Here we have our NADPH being created. And here we have our ATP synthetase. Well, remember when we looked at cyclic photophosphorylation, one of the things that this diagram really emphasizes is electrons have more energy at the beginning of this process than they do at the end. Okay, seems like a waste to let all that energy just, dissipate into the environment. So how can we avoid that? Well, as the electrons are passed around between all those electron transfer proteins, so here's plastoquinone, PQ, the B6F complex, plastocyanin. As the electrons are passed along, some of that energy that they lose is used to pump hydrogen. And the hydrogen is pumped out of the chloroplast stroma. Remember, that's the fluid in the chloroplast. And it's pumped inside the thylakoid into that thylakoid lumen. So hydrogen ions are pumped in here. And notice we also have some hydrogen ions because that oxygen evolving complex broke water into little pieces, and one of the little pieces was hydrogens with positive charges. So this is the inside of the thylakoid. So notice on the inside of the thylakoid in the lumen, I said you could compare our thylakoid to those foil-colored chocolate coins. This is in that chocolate area. We have hydrogens building up. And out here, we're pumping hydrogens out of here. So there's a lot of hydrogens in here in the thylakoid lumen. There aren't that many hydrogen ions. out here in the stroma. So in other words, there's a high concentration in the lumen, there's a low concentration out in the stroma. Well, what do we know about what happens when you have a high concentration next to a low concentration? Diffusion. Things move from where they're at a high concentration to a low concentration. So these hydrogen ions desperately need to get back out to the stroma. They need to equalize the concentrations. But there's a problem. If we look at this membrane, notice here's our two layers of phospholipids again, with the hydrophobic ends pointing out, the hydrophilic ends pointing into the middle. If we look at our plasma membrane is impermeable to hydrogen ions. So they can't get from the lumen to the stroma. So this means that the pressure is building up in the lumen. This thylakoid won't let the hydrogen out, and the hydrogen is kind of pushing on the membrane. It's trying to get out. This is where that molecule called ATP synthase comes in. ATP synthase, or it's sometimes called ATP synthetase, two names for the same thing, this will allow hydrogen to move back out to the stroma. So these dotted red lines show we've got hydrogen coming over here that got pumped in to the lumen. We've got hydrogen coming over here that came from the water that the oxygen evolving complex broke up. And this hydrogen is going to move through the middle of this ATP synthase molecule. It's one of those proteins that looks kind of like a channel. It's got a hollow tunnel through the middle. Now, since some of these hydrogens come from water, remember when water moves across a membrane by diffusion, we don't call it diffusion, we call it osmosis. Now, this isn't all of a water molecule, it's only the H pluses. So this process where we pump the hydrogens in and the hydrogens go back out, this is called chemiosmosis. Now, when the hydrogens go out, because there's so many inside, it's kind of like, one of those scenes where a crowd is trying to get out through a few doors. There's all this pushing and shoving. These hydrogens have a lot of energy. And when they go out, that energy is used by ATP synthase to take ADP and phosphate and combine them to make ATP. So this is how we actually make the ATP during cyclic photophosphorylation and non-cyclic. In both processes, around that PQ and B6F, we're going to pump hydrogen ions into the cell. We're going to get some more hydrogen ions by breaking water up into small pieces, and we're going to let those flow out through the ATP synthase. And when we do, we're going to make ATP. So let's think about overall, where our energy started out, where it went. If you think about this, if we go back to the beginning, we had energy in sunlight. That energy got transferred to electrons. Some of the energy from the electrons got transferred to the hydrogen ions, I'm sorry. From the hydrogen ions, that energy got transferred into ATP. Now, you might look at all this and think, why are you making it so complicated? Well, this is how it actually occurs. It might not be how you would design this if you were an engineer, but you have to keep in mind that when living things do metabolic pathways, a lot of times these pathways come from other pathways. So it's not necessarily what you might think is the most logical way to do things. So any questions right now about the light-dependent reactions? Okay, I'm going to stop screen sharing then. Tomorrow we will start out class. We'll have our cell test, unless I hear that, people are still having big issues accessing the diffusion problems. I tried, talked to our IT department and tried uploading them a different way, and I think you should be able to see them and submit them now. But we'll plan on having the cell test. You'll have an hour and 15 minutes to work on that. So that'll start at, we'll do the test at the beginning of class. So you'll have from 9 to 10:15. Like usual, the questions will be up there probably about 10 minutes early, I'm guessing, or so. This test is about the same number of questions as the chemistry test. So it will be a similar number of points. From now on out, all of our points will be somewhere between 150 points and 200 points. So chapter 1 test is kind
Can I
of
throw in
small.
a cell organ again today? Because I
Yeah.
was, what is that?
Yeah.
Okay. Because
So
I
yeah,
think
if you haven't turned in the organelles or you haven't turned in the diffusion problems yet, do those soon.
Okay, because I'm gonna
I
do
think
that.
it will help you on the test also if you have those questions answered.
Yeah. What if I don't understand what you're saying, class? Should I just read the notes? Because I like reading the notes, but at the same time, like
You can try reading the notes, but I mean, I would also recommend, either read our book or read another book, or you could go online. You have to be a little bit cautious if you go online because, anybody can post anything online, so it might not be accurate, but sometimes it will help if you get it from a different point of view. Now, another possibility is South Suburban College has an academic assistance center where there's free tutoring involved. And I haven't heard this summer, but I know most semesters they have somebody who works with the biology students.
yeah, I did
So,
it. Some of the teachers didn't know about what we was talking about, but a lot of them knew like most half of the stuff, like more than half the stuff.
yeah, I'm just thinking if you get it from a different perspective, that might help more. I mean, you can always email me and we can talk again, because if I know exactly what your question is, I can explain it a little bit differently to you than I do to the whole group. But sometimes a different perspective is good. So I don't want to discourage you from reviewing the notes, but also look at the book. Looking at the notes is good because I don't say every word that's in the notes. I add some stuff. I skip over some stuff that I figure you can understand because I assume you're going to read it. But the book is good because it has a slightly different perspective.
Okay.
Any other questions? Okay, if not, I will let you go today. So don't forget, check on those 2 assignments and